This is the Sakeology Center, Niigata University. The center is a hub for multifaceted studies on Japanese sake. Every day, a large number of students learn the new academic discipline, Sakeology. One of the most important and fundamental topics for us is the mechanism of sake fermentation. My research field is actually anatomy. My hobby is painting. So, illustrating the microscopic world is both my specialty and hobby. I'm here today to create a new piece based on the miraculous processes of fermentation. Well now, let our imagination run free and embark on a journey into the world of fermentation. The raw materials for sake brewing are rice and water. The inner layer of rice contains mainly starch. The outer layer contains proteins, fats, and many other components which are undesirable for taste and aroma of sake. Therefore, the outer layer of rice is polished off to create white rice, which is used for sake brewing. This is koji mold. Its scientific name is Aspergillus oryzae. It is a type of filamentous fungus and a microorganism that plays a crucial role in sake brewing. Spores of koji mold are inoculated over the surface of steamed rice. Once the spores germinate, they will extend their mycelia toward the inside or outside of the steamed rice. A sacharifying enzyme, amylase, essential for sake brewing, is secreted from the tips of the mycelia. The product of this stage is called kome koji or rice koji. Another microorganism essential for sake brewing is yeast. The scientific name of sake yeast is Saccharomyces cerevisiae. Since it proliferates by budding, it is also called budding yeast. Inside the tank for sake brewing are rice koji, water, steamed rice, and yeast to create sake mash, moromi. This is the starting point of sake fermentation. What is happening inside the sake mash? Let's begin our tour of the microworld, the beginning of a journey into the secret of sake brewing. The first thing we find in the sake mash is rice starch. Rice starch is a polymeric carbohydrate consisting of glucose units joined by glycoside linkage. Inside the sake mash, the starch is hydrolyzed to glucose with the help of the sacharifying enzyme amylase produced by koji mold. This process is called Saccharification. Inside the yeast cells, we can overlook glucose, ethanol, and carbon dioxide, which originated from saccharides formed via starch saccharification. Yeast is similar to humans, a eukaryote with a nucleus. For that reason, they have various organelles, just like human cells. This is the glucose transporter of a yeast cell. It is a protein found inside a lipid bilayer, a type of cell membrane, and it transports monosaccharides into the cell. This is how glucose enters the cell. Let's go into the yeast cell together with the glucose. Now we are inside a yeast cell. From a single molecule of glucose transported into the cell, a multi-step reaction of 11 enzymes in the cytosol produce pyruvate as an intermediate, and finally, 2 molecules of ethanol and 2 molecules of carbon dioxide. Let's look at this important pathway for energy production called the glycolysis, where glucose is converted to pyruvate. First, glucose 6-phosphate is generated by the phosphorylation of glucose on the 6th carbon atom. Let's run through to where the pyruvate is generated. Pyruvate, a key intermediate for ethanol production, is generated from phospholenol pyruvate, completing the glycolysis. After this, we'll finally observe ethanol generation by a two-step enzyme reaction. First, as you can see, carbon dioxide and acetaldehyde are generated from pyruvate. Now, we move on to the final stage. Ethanol is produced from acetaldehyde, which completes the alcohol fermentation. Till this point, we've observed the saccharification and alcohol fermentation processes. Saccharification is the process in which amylase, produced by koji mold, helps degradation of starch into glucose. Alcohol fermentation is the process in which glucose is transported into yeast cells and finally converted into ethanol and carbon dioxide. These two processes occur simultaneously in the sake mash. This is called multiple parallel fermentation. Sake is brewed using this unique process. This concludes our sake brewing microworld tour. An epic story in a micro world. I hope you'll remember this tiny world the next time you enjoy sake.